Hello everyone, I am Bhaskar, Assistant Professor of Department of ECE, HiSET. In this video, we are going to discuss the elements of visual perception in the subject Digital Image Processing. The contents are the structure of human eye and image formation in the human eye. These are the two topics we are going to discuss in this video. Basic objective of image processing application is to help human observer to perceive the information from the visual information or an image. So it is important to understand the basic functionality and structure of our human eye. In our visual system, we have three parts. The first actually starts from the eye, which is acting as a sensor or camera. Optic nerves, which transmit the information to brain. Then the third part is brain, which is actually acting as a CPU, central processing unit, where the actual information is interpreted. There are three different membranes in the human eye. Those membranes are outer membrane, which consists of cornea and sclera, then middle layer, choroid, then inner layer is retina. Eye is almost a spherical, almost in spherical structure with diameter of 20 mm. The outer layer, the outer transparent layer, transparent segment of the front portion, anterior portion is called as cornea. This portion. This is transparent in nature. So the same cornea is extended in the rear as sclera. So in the rear, it is opaque in nature. It won't allow any light to get past. The middle layer is choroid, where iris, then ciliary bodies, and iris diaphragm, lens, all those things are present. In the inner layer, we have photoreceptors like cones, rods, and blind spots. So these are the three basic membranes. In choroid, we have blood vessels. So these blood vessels, in the middle layer, we have blood vessels which provides nutrition to the muscles of this eye. If we get injured, the eye becomes red. That reddishness is because of the damaged blood vessels in the choroid. Next is iris. Iris is a part of this choroid. Actually, this iris will give us the color pigments. We have different colors in the eye: brown, black, green, blue. All these color pigments are because of this iris. Then we have pupil. The center opening of this iris is called pupil. This pupil controls the amount of light entering to the eye. Pupil size is around 2 to 8 mm. Then we have lens. Then we have lens of this eye, which is made up of fibrous cells containing water, 60 to 70% of water. Then the innermost layer is retina, where the different photoreceptors are present. The photoreceptors are cones and rods. These two photoreceptors have different characteristics as well as different functionalities. First, cones. There are around 6 to 7 million cones are present in the retina. Then all these cones are accumulated in one place, which is called as fovea. So these cones are responsible for bright light vision. Bright light vision is otherwise called as photopic vision, where all the color information can be perceived. Similarly, these cones are responsible for fine details. The high frequency information can be perceived by these cones. Then rods, there are around 75 to 150 million rods are present in the retina. They are distributed all through this retina, and these rods are responsible for dim light vision, otherwise called as scotopic vision. This dim light vision, it will not give any details as well as color information. Fovea is the central part of the retina. It is about 1.5 mm diameter, highly dense with cones. Below this fovea, we have a blind spot where there is no receptor present. Now, it is important to understand the difference between photopic vision and scotopic vision. Photopic vision is otherwise called as bright light vision. Scotopic vision is dim light vision. Now, the characteristics of photopic vision are the cones are responsible for this photopic vision. Then color receptors, the color discrimination is high, the resolution is high in the fovea, that is in bright light vision, and this is less sensitive to light. When we look into the characteristics of scotopic vision, rods are responsible for this scotopic vision. There will not be any color discrimination, and it is much more sensitive to light, that is night vision, and it will not give any details, that is the resolution is low in this scotopic vision. So this is how actually the photoreceptors are distributed along the retina. So if we look at this, 0 degree represents exactly the visual axis. Visual axis, if you look at the structure, we have a dotted line. This is called visual axis. So the fovea is exactly at the 0 degree location. In the fovea, we can see that the number of cones are high, very high. That is, cones are densely populated in fovea. And in all the other places, the number of cones are very less. But whereas the rods, the dotted line represents the number of dots, the continuous line represents cones, number of cones. Okay, so if we see in the fovea, the number of cones are highly tensed. Rods are distributed throughout the retina. But in one place, we don't have both rods and cones, which is called as blind spot, which is somewhat below the fovea. So the dimension of fovea is 1.5 mm, and fovea is rod free. There will not be any rods, only cones are accumulated there. So these are the photoreceptors of the eye. So this is the basic structure of human eye. Next is image formation in human eye. Actually, to perceive any object, we need a light source. Naturally, the light source in the daytime is sunlight. So if the sunlight falls on any object, those light reflected from that object and it reaches our eye, then our eye captures those light waves, then it transmitted to the brain through these optic nerves. Then in the brain, all these interpretations are happening. To focus a distant object, the lens must be flattened, the focal length must be higher. To focus a nearer object, the focal length will be less. So the size or the shape of the lens must be changed. So for that, ciliary bodies are changing the focal length of the lens by controlling the thickness and the shape of our human lens. So this is where the nearsighted and farsighted perceptions are coming. So this is how image is formed inside our eye. Actually, if we have the length, the distance between the center of lens and fovea is around 1.7 centimeters or 17 mm. If we look at a tree with a height of 15 meters at a distance of 100 meters, then an image is formed in the fovea, and the size of that image can be measured by this formula. That is height divided by distance. 15 by 100 equals to height in fovea. Height in fovea divided by this distance 17 mm, which is approximately 2.5 mm. So this is how the image is formed inside the eye. In this short video, we have discussed the basic information about the structure of human eye and how the image is formed inside human eye. Thank you.